Good afternoon, everyone. I'm so glad to have you all here. We are the MyMark team. So, breast cancer is the most common cancer for women, so it's a big problem. 1 in 8 women will develop breast cancer in their lifetime. My mom is actually a survivor, so that's why I'm very concerned that 1 in 5 mammograms fail to detect a breast cancer or produce a false negative. This is a problem, but not for much longer. Introducing MyMark, the inexpensive, non-invasive, easy point-of-care early detection method for breast cancer. You only have to remember 3 things: spit, drop, and read. You spit into a cup, you drop the spit onto our microchip here, and you wait 15 minutes for the app to announce your result of whether or not your biomarkers are within the abnormal range.
There are certain salivary biomarkers shown to be elevated in women with breast cancer. Because we want to further research, we can use our modular device to test other biomarkers in the 5th slot. So the way that it works is that our lab on a chip detects information about the biomarker concentrations and sends it to an application, which will determine if it's within the normal range. Then, if the levels are elevated, the patient will be recommended to see a provider. Then, the patient will enter a unique code back into the application to give us some feedback and answer a quick survey to further research.
All right, let's take a look at a demo and see how this works. So, first, the patient will open up our MyMark app, which the team built this weekend, and they'll hit Start. Now, the first step is to complete an informed consent process that is integrated with Apple's Research Kit. Once the patient agrees, they provide their saliva sample on the device, which relays the signal, our lab on a chip, which relays the signal back to the iPhone for analysis. Now, while the patient waits for their result, we ask them to complete a brief survey, which we're going to skip here in the interest of time, and then finally, we display the preliminary result along with a request to the doctor to send us feedback using a unique but memorable code. All right, let's talk business plan. Stage 1 is all about data collection. So we will partner with a group of radiologists who agree to use our test as part of their routine annual mammogram screenings with cancer patients. Using the data that we collect in stage 1, we will refine our machine learning and predictive models so that we can better predict early cancer. Now, we will segue into a clinical trial for FDA approval, and once approved, we will begin the full-scale production rollout. This non-invasive test can be used to enable the early detection of cancer at a fraction of the price of today's more invasive alternatives. And at this price point, we can enable women around the world, including many in countries with little to no access to early screening today, to detect cancer early while it's still treatable. And that's a vision that our team believes in. With that, thank you very much, and we look forward to answering your questions.
That's a great presentation. The problem with cancer diagnostics is that the sensitivity is usually such, even in serum, that you're picking up late-stage disease. And so one question here is, can you actually do that in saliva? And why breast? Why not lung? Lung would seem to be a much better candidate for a saliva test. And then 2, what would be the legal impacts of having false negative or false positive data and having follow-up? And how do you think about addressing that?
Okay, so we will divide that question up. First, we do have biomarkers, and we have the thresholds that they detect, as well as the sensitivity, which are upwards in the 80% range. And then you also asked about lung cancer versus breast cancer. I understand what you're saying because it's in saliva. However, there have been multiple studies that show that these same biomarkers are present in saliva, and we wanted to address something as prevalent as breast cancer.
The other thing that I would add here is that part of the chip that we developed this weekend, the thought here, and getting to business model, is that we are actually developing a platform that is scalable for adding other biomarker detection to these same kind of chips that can be manufactured cheaply and at scale. So in the future, if we want to branch out to other types of cancer, we have the infrastructure and the platform that will enable us to do that at scale.
The biomarkers that you've chosen, if they are consistently expressed or present in saliva at levels that can be detected and are not changed by eating or what have you, or other activities, why isn't there a test already?
Well, currently we have been, all the literature. Hi, everyone. So the current literature review basically shows that most of these tests are serum-based, that is blood tests rather than saliva. The problem was analysis of saliva. So with the microfluidic chip, now we actually have a tool to replicate a lab ELISA test at home on chip, which can be linked to your computer. So although it could have been done, we just didn't have the technology to do it.
And these microfluidic chips have already been published on in the literature, but separately. So what we're doing here is we're multiplexing the research that already exists and packaging it as a breast cancer diagnostic or screening tool, rather than just an individual chip that's just used by a couple of researchers.
Thank you.
Thank you.